Okay, so like imagine you're a detective, right? And you're trying to find this tiny little clue at a crime scene. You need a tool that's like incredibly sensitive, you know, and really specific. That's kind of what ELISA is in the world of molecules.
Yeah.
We're going deep into ELISA controls today,
Okay.
and specifically like why they're so crucial for accurate results.
Right.
You've done some ELISA work before, I think you said.
Yeah, yeah, definitely. Quite a bit, actually.
So you're probably pretty familiar with like how antibodies bind to antigens and all that.
For sure. Yeah.
But like, have you ever thought about all the things that could like go wrong in that process?
Oh, absolutely. I mean, there are so many places where things can go sideways.
Right.
It's kind of like baking a cake, you know?
Hmm.
You can have all the best ingredients and the perfect recipe, but if your oven temperature's off or you forget like the baking powder, the whole thing's going to just totally flop.
Exactly. And that's where these controls come in. They're like the checks and balances that throughout the process. Let's start with the like positive and negative controls.
Mhm.
Those are like the foundation of a good ELISA experiment.
Right. Absolutely. They're like your sanity checks,
Mm.
you know? The positive control, it's like a litmus test. It's a sample where you know for sure it contains that target that you're trying to find. So if the assay is working like it should, you should get a nice, strong signal from that positive control.
So it's not just about like seeing if something's there, but it's also about confirming that the entire process is like working the way it's supposed to. What happens if that positive control doesn't give you that expected result?
Oh, that's a huge red flag. That could mean your reagents have gone bad, or maybe there's a problem with like your procedure. It basically tells you to stop everything and figure out what's going on before you even look at your actual samples.
Got it. And the negative control is kind of like the opposite, right? It helps you rule out those like false alarms.
Precisely. Yeah.
Yeah.
The negative control should be totally free of whatever that target is. If you start seeing a signal there, it means something's interfering with your assay. Maybe there's contamination or some non-specific binding happening.
So the negative control is like your baseline. It helps you tell the difference between like a real signal and just background noise. Okay, let's move on to something I think is a little more complex, standards. From what I understand, these are really important when you need to know not just if something's present, but like how much of it is there.
You're exactly right. Standards are like your ruler for ELISA. They give you a series of known concentrations of your target, and you run those right alongside your unknown samples.
But how do you use those to like actually figure out the concentration of your unknown?
That's where it gets really cool. When you plot the signal from your standards against their known concentrations, it creates this calibration curve. And this curve is like your decoder ring, right? It lets you translate the signal from your unknown sample into an actual specific concentration.
Ah, that makes sense. So it's like you've got this map that guides you from the signal intensity to like the actual quantity of your target.
Exactly.
Pretty clever. What about when you're dealing with like more complex samples like blood or tissue samples? I imagine there could be all sorts of things in those samples that could mess with the ELISA.
Oh, for sure. You are totally right. Real-world samples can be really messy. And that's where these spike and recovery controls come in. Imagine you're trying to measure like the caffeine in a cup of coffee.
Okay.
You wouldn't just add your ELISA reagents right into that coffee, would you?
Probably not.
No, because there's all sorts of other things in there that could totally interfere. So
Uh-huh.
you spike that coffee with a known amount of pure caffeine,
Okay.
and then you see how much of it you can recover using your ELISA.
So it's like a test run, but in a more like realistic setting?
Yeah, exactly. Spike and recovery tells you how well that assay actually performs in that complex sample and whether or not all those other components are like messing with your measurement.
Okay, that makes a lot of sense. So we've got controls to make sure the ELISA is working, controls to actually quantify our results, and controls to check for like interference in those complex samples. What else could possibly go wrong?
Well, sometimes you have to dilute your samples, like if they're super concentrated.
Okay.
And that's where these linearity of dilution controls come in.
So you're basically checking to see if the ELISA still behaves itself even when the sample is diluted.
Exactly. So you make a bunch of dilutions of a known sample, and you run them all through the ELISA. If everything's working right, you should see this nice proportional decrease in the signal as that concentration goes down.
So it's like making sure the ELISA is playing by the rules across like a whole range of concentrations.
Exactly. Now imagine you've got this sample, and it's like way off the charts. It's way above the highest standard on your calibration curve. You'd have to dilute it to get a measurement you can actually trust. But if the assay isn't linear across those dilutions, you could end up with results that are just completely wrong.
Ah, I see. So linearity of dilution is about making sure that the assay is reliable, even when you have to adjust the concentration of your sample.
Okay, that's fascinating. I never realized how much thought and planning goes into designing these controls.
It really speaks to like the power of the scientific method and how important this rigorous testing is. Trust me, these little details, they can make all the difference in like the reliability and accuracy of your ELISA results.
I'm starting to see that. So we've covered the positive and negative controls, standards,
Yeah.
spike and recovery, linearity of dilution. It's like we've assembled this whole toolkit to make sure our ELISA experiment is like really robust and trustworthy.
Exactly. And we haven't even gotten to blanks yet. Those are another super important piece of this whole puzzle.
Oh, wait.
But maybe we can save those for the next part of our deep dive.
Yeah, let's do that. We'll pick up right where we left off, and I have a feeling things are going to get even more interesting, for sure. It's kind of funny, right? Like, we always focus on those exciting results of an experiment, but the real heroes are often like those behind-the-scenes controls, quietly making sure that everything is, like, running smoothly.
Yeah. They're like the stage crew in a theater production, you know? They don't get the standing ovation, but without them, the show couldn't go on.
Exactly.
So you mentioned blanks before. What role do they play in this whole ELISA performance?
Blanks are all about, like, setting the stage for those accurate measurements. It's like zeroing out a scale before you weigh something. They basically help us account for any of that background signal that's not actually coming from our target.
Okay, so it's about like eliminating any interference that could skew our results. Are there different types of blanks that we need to think about?
Yeah, so there are two main types, sample blanks and reagent blanks.
Mm-hmm.
The sample blank is specific to, well, your sample. It accounts for any absorbance that might come from the sample itself, like not the target that we're interested in.
So it's like taking a snapshot of that background noise that's like inherent to that particular sample. Yeah.
Okay, that makes sense. What about the reagent blank? How is that different?
The region blank focuses specifically on the regions that you're using in that ELISA, especially a secondary antibody. It helps us spot any non-specific binding of that antibody, meaning it's sticking to things that it shouldn't.
Ah, so it's like making sure that the secondary antibody is like behaving itself and only binding to the primary antibody, which in turn is bound to our target.
Right.
If we see a signal in that region blank, that's a sign that something's not right.
Exactly. It could mean that there's some contamination in our reagents or that secondary antibody isn't quite as specific as we thought it was.
Right.
And that can lead to those false positive results.
Wow. I'm really starting to appreciate how each of these controls like addresses a specific challenge or potential pitfall in this whole ELISA process. It's like they've anticipated every way that things could go wrong, and they've given us these safeguards to like keep us on track. Yeah, that's a really good way to put it. And when you think about like the applications of ELISA in diagnostics and drug development and all sorts of research, those safeguards become even more crucial.
Absolutely. Speaking of those real-world applications, you mentioned earlier that ELISA is used in both research and diagnostics. Can you give us like some concrete examples of how these controls actually play out in those different contexts?
Sure. So let's start with diagnostics. Imagine you're using ELISA to test for like HIV antibodies in a patient's blood sample.
Okay, so in this case, we're looking for the presence or absence of a specific antibody, which would indicate whether or not that person's been exposed to the virus.
Exactly. Now, that positive control in this situation is super crucial. It's a sample that we know for a fact contains HIV antibodies. So if the ELISA is working the way it should, this control should give us this strong, clear signal.
So it's not just about detecting the antibody in the patient's sample, it's about confirming that the test itself is actually capable of detecting it. What if that positive control fails?
That's a big problem. That could mean that the reagents have gone bad, or maybe the equipment's malfunctioning, or there's an error in the procedure. It basically invalidates that entire batch of tests.
Wow, so the positive control is like the gatekeeper? It's making
Yeah.
sure that the test is reliable before we even look at any patient samples?
Exactly.
What about the negative control in this situation?
So the negative control would be a sample that we know does not contain those HIV antibodies, and this helps us rule out any false positives. If we see a signal in that negative control, it means something's interfering with our assay.
Right, because a false positive in this context could have really serious consequences. It could lead to a misdiagnosis, unnecessary anxiety, and even like inappropriate treatment.
Yeah, exactly. So in a diagnostic setting, those positive and negative controls are just absolutely essential for making sure we get those accurate and reliable results, which of course directly impact patient care.
That's a powerful reminder of like what's at stake here. Okay, so we've seen how those controls play out in diagnostics. What about like a research setting?
Okay, so let's say some researchers are studying the effects of like a new drug on a particular protein marker in some cells, and they're using ELISA to quantify the levels of this protein in cells that have been treated with the drug compared to cells that haven't been treated.
Okay, so they're looking for changes in protein levels as like an indicator of that drug's effect.
Right.
In this case, would the standards be particularly important?
Oh, absolutely. Remember, those standards are those known concentrations of our target protein that we run alongside our unknown samples.
Right. They help us create that calibration curve, which is like our translator between the signal intensity and the actual amount of that protein.
Exactly. In this research scenario, those standards allow the researchers to like very precisely quantify the protein levels in both the treated and the untreated cells.
Okay.
And that way they can determine if there's a real statistically significant difference between those two groups.
So the standards are providing that like quantitative framework for comparing those groups and drawing conclusions about the drug's effect. What about the other controls in this research context?
Well, the positive control would make sure that the ELISA is working correctly and that it can actually detect that protein in the first place.
Mm.
And the negative control would help to rule out any false positives, for example, if there was some other protein in those cells that was kind of cross-reacting with the ELISA regions.
Okay, that makes sense. But what about those more specialized controls we were talking about earlier, like spike and recovery and linearity of dilution? Would those come into play in this kind of research?
Oh, they definitely could. Like if the researchers are working with cell lysates, those are complex mixtures of proteins and other cellular components. Spike and recovery would be super important in that case. It would tell them how well the ELISA performs in that messy environment and whether or not all those other components are interfering with their protein measurement.
So it's like a reality check to make sure that the ELISA is still accurate, even when things get really complicated.
Exactly.
And what about linearity of dilution?
Linearity of dilution would be really important if those researchers needed to dilute their samples. For example, if some of those samples have protein levels that are just off the charts, like way above the highest standard on that calibration curve, they'd need to dilute them to get a measurement that they could actually trust.
Right, because if the assay's not linear across those dilutions, those diluted samples could give them results that are just completely off.
Yeah,
Exactly. So linearity of dilution is about making sure that the ELISA is playing by the rules across a range of concentrations, which is especially important when dealing with those samples that might need to be adjusted.
You got it. And this highlights a really key point. The choice of controls depends a lot on the specific experiment and the types of samples that you're analyzing.
Right.
There's no one-size-fits-all approach here.
Okay, that makes sense. It's about tailoring those controls to the specific needs or potential challenges of each experiment. But in all cases, these controls are like the unsung heroes working behind the scenes to make sure that the data is accurate, reliable, and trustworthy.
Exactly. And that's why it's so important for researchers to understand not just the how of running an ELISA, but the why behind each of those controls and how it contributes to the overall validity of those results.
It's like the difference between following a recipe and actually understanding the science of baking.
I love that analogy. It's about having that deeper understanding that lets you troubleshoot and adapt and ultimately have confidence in those findings.
Speaking of confidence, I'm feeling a lot more confident about my grasp of ELISA controls now.
You too.
We've covered so much ground, from like those basic principles to those real-world applications in research and diagnostics.
It's been a great deep dive, but I think it's time to shift gears a little bit and explore a slightly different angle. We've talked a lot about how the absence or misuse of controls can cause problems. But what about like the flip side of that? Could there be such a thing as too much control in ELISA or in science more broadly?
Ooh. That's an interesting question. It's like that age-old debate about like the balance between regulation and innovation.
Exactly. Is there a point where too much control actually hinders scientific progress? That's something I'd love to explore further.
Well, I'm definitely intrigued, and I have a feeling our listeners are too.
Let's take a short break, and when we come back, we'll delve into this really fascinating topic of finding that right balance between control and creativity in that pursuit of scientific knowledge.
So we've spent like a lot of time talking about the importance of controls in ELISA and how they're like those safeguards against errors and misinterpretations. But now I'm thinking, could there be a downside to being like too careful, too controlled in our experiments?
It is a fascinating question because on the one hand, we want to be super rigorous, right? and make sure those results are rock solid.
Hmm.
But on the other hand, science is also about exploration and pushing those boundaries, sometimes even taking risks.
Hmm.
Too much control could stifle that whole spirit of discovery.
I see what you mean. It's like trying to be too precise when you're like sketching out an idea. You might end up with a drawing that's technically perfect, but it could lack that spontaneity and creativity of just like a rough draft.
Exactly. Sometimes those like messy explorations lead to the most unexpected breakthroughs. If we're so focused on like controlling every single variable,
Mm.
we might miss those serendipitous discoveries.
So how do we find that balance? How do we make sure we're being rigorous, but we're not like stifling innovation?
Well, I think it starts with recognizing that those controls are tools, not these like rigid rules.
Okay.
We need to be really thoughtful about which controls are like truly necessary for a particular experiment
Yeah.
and which ones might be adding more burden than benefit.
So it's about being strategic and selective with our controls rather than just like throwing everything at the problem.
Exactly. And it's also about being open to like trying new approaches and not being afraid to question those established protocols. Sometimes a fresh perspective can lead to controls that are more efficient and effective, but still maintain that scientific rigor.
Makes a lot of sense. It's about finding that sweet spot where we have enough control to ensure that our data is good, but not so much that it like stifles our creativity and ability to explore new ideas.
Absolutely. And I think this applies not just to ELISA, but to science in general. We need to strike that balance between like standardization and flexibility, between following those established methods and being willing to challenge them when we need to.
It's a constant evolution, isn't it? Science is always moving forward, and our approaches to controls need to like evolve right along with it.
Precisely. And this brings us back to something we talked about earlier, the importance of understanding like the why behind each step in that scientific process, including the controls. When we really get those principles, we can make better decisions about which controls are essential, which ones can be adapted, and which ones we might not even need.
It's like being a chef who not only knows how to follow a recipe, but also understands the chemistry behind why certain ingredients work together. That deeper understanding leads to more creativity and innovation in the kitchen.
Exactly. Same goes for science. When we go beyond just memorizing stuff and really embrace those underlying principles, we empower ourselves to be more creative and adaptable and ultimately more successful in our scientific endeavors.
This has been like such an eye-opening conversation. I feel like I've gained a whole new perspective on ELISA controls and on scientific research in general.
It's been a pleasure diving into this with you, and I hope our listeners are walking away with a similar sense of curiosity and a deeper appreciation for the nuances of scientific methodology.
I'm sure they are. So until next time, keep exploring, keep questioning, and remember that even in the world of science, sometimes a little less control can lead to a lot more discovery.